interesting talk. Our next speaker is Jacob Robinson. He is, and Mesa, make sure you stop sharing. Jacob is an associate professor in ECE at Rice University. He got his PhD from Cornell, was a postdoc in Department of Chemistry and Chemical Biology at Harvard University. And during that time, he developed electrical and chemical silicon nanowire devices to probe electrical and chemical activity of living cells. And his lab at Rice is really interested in developing nanofabricated devices to study structural and functional dynamics of living neural cells, circuits. He is the 2014 DARPA Young Faculty awardee, is the Dunn Award recipient, and his talk today is about magnetic and optical neural interfaces. The floor is all yours.
All right, now can you hear me? Well, I'm gonna assume yes, because my microphone's moving up and down. Okay, yes, all right, that sounds like yes. Thanks, Yana, and all the org. Actually, I share Cindy's sentiment that I really wish I could have visited in person, because I always love to chat with you guys while I'm there. But hopefully we'll have opportunities over Slack and the Q&A. Let's see. So I realize, looking at the program, that I'm in the optics section, but I'm gonna go rogue a little bit and talk a lot about magnetics. So my apologies, although there is going to be a little bit of optics at the end. I guess I don't know. I can make a bad joke that magnetics is also uses photons as the force carrying particle. So in some sense, this is all optics, right? We'll try to go with that. Okay. So first of all, everything I'm talking about today by this fantastic group of people, and I'll mention them as we go along. I won't dive too much into this motivation, because I think we all share the same kind of vision that we'll be able to connect the body to computers somehow to treat a number of medical neurological disorders, maybe one day connect to our, yes. So I can, but what I've done, what I found in the past is that my videos don't play very well if I do that. So I'm going to do this. Okay. If it's okay, I'll do it in this, and then
it's okay.
Yeah, apologies. So I'll have to pretend like that thing on the side isn't there. It's only for me. Okay. Let's see. So yeah, and I guess we also like to imagine that, you know, if we do a really good job of making these devices, if they're super non-invasive and really high performance, then maybe there'll be the opportunity to expand into like a consumer electronics market, and we can, like, you know, plug our brains into computers, which would be fantastic. Okay. I tend to think about, given a background in physics, is this transduction problem. So we have information carried by ions and chemicals inside the body. Carried by ions and chemicals inside the body. We're trying to get that information into the form of electrons that move around in hard two-dimensional silicon circuits. And so I think about what are the technologies we can use to convert between biological signals and electronic signals. This is not the only problem in developing these neural interfaces. It's just the one that I happen to be most interested in. What our thinking has been, and where we've been thinking a lot of opportunities lie, is ways that we might be able to miniaturize devices so that they're either injectable or implantable at the millimeter scale, so very, very tiny. And they have great chemical specificity and the ability to communicate over relatively long ranges, so that we don't have to have tethers. Basically, I want untethered technologies that have good biochemical specificity. A lot of this has led us to the idea that these technologies might combine some elements of synthetic biology, where we either genetically modify the cells or introduce genetically modified exogenous cells into the system, because biology has already solved the specificity problem, right? We have receptors in the body that are very sensitive to specific neurotransmitters. Doing this artificially turns out to be really hard in an in vivo system where there's potential for biofouling. So I think there's a lot of opportunity to leverage what nature has already solved in terms of chemical specificity. Combine that with nanotech, which is, as you heard from Flavia and Zahi and a number of other people, and Cindy earlier today, does a really great job of this bioelectronic transduction. So I'm not gonna talk a whole lot about this, but I'm just giving you a sense of like where I think we're heading. And I think it's this combination of bioelectronic signal transduction with chemical specificity from synthetic biology. Okay, so why do I want these little tiny implantable guys? So when we think about the opportunities for bioelectronics, both for neural interface as well as for bioelectronic medicine, there are a number of targets. They're distributed throughout the body. For example, you know, a number of deep brain targets, cortical targets, maybe you want peripheral nerve stimulators and things that might measure activity in the periphery. And in general, I imagine this network, this kind of distributed biological electronic network of sensors and actuators that allow me to develop closed loop control systems to regulate physiological processes, control levels of pain, control mood, attention, develop prosthetics, et cetera. And so I don't think that these are going to be supported by large devices that have a battery pack and a central pattern generator connected to a lead. And even if you miniaturize this battery powered devices, they're still relatively large and don't have a lot of communication capabilities. So I think we want to realize this future of this kind of distributed bionet. We're going to have two solutions that have wireless. Oh my God, I'm sorry. I'm getting a lot of Slack messages. I need to figure out how to mute this. Okay, so we're going to need to develop this system of wireless modes, I think, right? So I'll talk a little bit about things like neural dust, which was mentioned earlier today. But basically, I'm thinking devices that can fit on the tip of your finger, sense, actuate, and be completely tetherless. Now, to do this, I think we need to develop a good wireless power solution for these types of things. So I'll tell you what we've been working on in terms of solving this wireless power for miniaturized devices. And the idea is by using magnetic fields to power these devices. And the reason why is that magnetic fields do an excellent job of penetrating in the body. Mason talked about the idea that light has a scattering problem, but you can solve this by focusing it. Magnetics, on the other hand, really don't interact very strongly with the body. So we don't suffer from reflections and scattering and that kind of stuff. And in order to convert this magnetic field that doesn't really interact with the body into something that's useful, we've come up with this idea of making magnetoelectric materials that power our bioelectronic devices. Okay, so how does this work? How can I make magnetic field to create useful energy for my devices? It works with a combination of two materials. One material is a piezoelectric material, which you probably know if you apply stress or strain to a piezoelectric, you'll create a voltage across that film. A magnetostrictive material, on the other hand, responds to magnetic fields by changing its size. It creates a small amount of strain in the film when I apply a magnetic field. So if I couple these two things together, if I turn on a magnetic field, the strain that is developed in this magnetostrictive film will be transferred to the piezoelectric, and the strain in the piezoelectric creates a voltage. So that's great. It's a way to convert directly a magnetic field into a local voltage. Now this effect is relatively weak, but if I actuate these devices, basically turn the field on and off at the mechanical resonance, it's an ultrasound resonance, it's an acoustic mode. At the ultrasound resonance of this device, I can get a relatively large voltage across it and transfer a fair amount of power. And so that's the principle. This was the idea that actually we had when Amanda Singer joined the lab about 5 years ago. She was an applied physics student and had some experience with magnetoelectrics. Hey, this is maybe can work. And over the last 5 years or so, she has really pioneered going from fabricating the films, putting them together with bioelectronic stimulators and actually demonstrating in vivo stuff. So huge hats off to Amanda and Josh and Kaiwan, who come on later to the project to add integrated circuits. Okay, that's it. That's basically what we're doing. We're gonna spend the rest of the time telling you kind of why I think this is important. Okay. So why did we go through all this effort to create a new material system to transfer energy? Well, this is my kind of, the best I could do at a survey of what people are using to power these miniature bioelectronic devices. Again, battery-free, leadless, the kind of things that we think is important for distributed systems. All right, so we basically have electromagnetic waves as one form of energy transfer. For these to be efficient, we need to make our devices about the size of the electromagnetic wave, right? So lambda EM here. Now, it turns out that these waves are usually relatively large. And so if I want to make a miniature sized device, something on the order of millimeters or so, I have to push that frequency up to gigahertz range. And at those frequencies, the body absorbs that energy pretty efficiently. And so these devices tend to only work at the surface. And you can use some clever techniques to kind of focus that energy into the body so that you can deliver power without creating too much heat. That's what's called midfield technologies, and it's a really fantastic approach. They're really fighting fundamental physics here because the devices have to be wavelength scaled in order to be efficient. So a super clever idea that was really pioneered by Michelle Maharvitz and Jose Carmena at Berkeley was to say, hey, look, I can make devices that are also resonant, but resonate with an ultrasound wave. The wavelength is much smaller. In fact, it's 5 orders of magnitude smaller because sound travels much more slowly than light. And so they created neural dust, which is basically these piezoelectric crystals that vibrate in response to ultrasound. So what that meant is that they could go from kind of these larger devices and really miniaturize them. So I'm kind of plotting, this is okay, the best I can to kind of collapse many different design characteristics into a 2-dimensional plot, so bear with me. But basically they took what was these relatively large devices and made them super small. Now the trade-off is that ultrasound waves don't propagate well from air to tissue. There's this huge impedance mismatch, and that's why you have ultrasound gel. Anytime you want to have kind of ultrasound imaging. Also, you tend to have to really focus your beam to your device, and if there's a slight angular or translational misalignment, then your coupling efficiency drops pretty dramatically. So that's why I kind of added this other axis here, which is robust performance. So although there's a huge win in making things small, we sacrifice what I call, this is kind of like pain in the assiness. So if you're all the way over here, using this stuff is a real pain in the ass because you have to have ultrasound gel, you have to have, great alignment. In some applications, that's fine. That's great. In other applications, if I think about, someone who's like running around or whatever, and you want to power your device, maybe that's, you want to be more robust. And that's why, wireless techniques or near field, which is down here, tend to be a little bit more robust. Okay, so let me talk about near field. So near field works by generating a magnetic field with a coil, and I have a pickup coil over here that captures a little bit of that magnetic flux, and I use it to create a current. So this is magnetic induction, and that's actually probably the most common wireless power transfer technology, and there's a reason why. The reason why is actually it gets very efficient, particularly as you create larger coils. So it's actually quite robust, but it's really tough to make these things tiny because as you make these pickup coils smaller and smaller, the area that you use to pick up that magnetic flux gets smaller and smaller. So you really don't, it's very, very hard to get devices kind of below this 10 millimeter threshold. Now, why is this 10 millimeter threshold interesting and or important? It's at this scale where we start to think about devices that are injectable, that can fit through the needle of a syringe, that can maybe even be introduced endovascularly on stents. And so I think the technologies that are kind of above this line, super exciting for this kind of distributed network. Below this line, it's a little bit harder to do that through syringe tips and that kind of thing. So the idea that we had, and I guess where I think magnetoelectrics fits in is it's kind of a combination of magnetic induction and ultrasound or these neural dust types approaches. And that's because we're using the same idea. We're basically vibrating a piezo crystal just like you do with ultrasound. But rather than using an ultrasound wave to drive it, we're using magnetic field. So we benefit from this idea that magnetics propagates through air and has no reflections at tissue air interfaces, goes through bone very well also. So we get all of those benefits of kind of the robust performance that we get from a near field link, but we get the really nice scaling properties that we get from ultrasound. So I guess this is the learning slide. So maybe screenshot this or whatever. So I think that this is basically the way that I think about the space. And so magnetoelectric still is a niche. It doesn't do everything well, but what it does well is I think it works well with this kind of non-contact. So if I have a coil that's at a distance from my from the body, for example, maybe in some kind of a wearable thing. That's where it's actually very nice, and only really for tiny stuff. If you want to make something big, you're probably going to use RF with the surface coil, or you're going to use near field because you have the space to make a large pickup. So in this niche for kind of injectable endovascular non-contact stuff, I think magnetoelectrics has a special place. Okay, so let me show you a little bit more of the details. I mentioned this idea that it scales very nicely. So there's a fundamental difference between the way that energy is transferred to a coil and energy is transferred to one of these magnetoelectric crystals. And that is, as I mentioned, as I make the area of these coils smaller and smaller, I'm transferring less power, and the voltage actually drops, falls off, like the area cubed, actually, because there's an inductance term here. But basically it falls off pretty dramatically. So that's why big coils are awesome, right? That's how you charge your cell phone. It's how you charge your Tesla or whatever. But as you get really small, performance really drops off. This is fundamentally different than the scaling properties of magnetoelectrics. And that's because the voltage depends on the strain of that film. So if I apply the same strain, I'm always creating the same voltage. I make my film smaller and smaller and smaller, the voltage stays the same. Power drops a little bit, but it falls off linearly with area, not at this cubic term. So this is, I think, really what's special. So again, small devices is where magnetoelectrics shines. There's another thing that it does really well too, and I'll play this video. It has really great angular sensitivity, which is one of the challenges, I think, with neural dust and with near field coupling. The reason why we get great angular sensitivity is the magnetic properties of the magnetostrictive material focus the magnetic field through the core. And so if I plot normalized power as a function of angular rotation, it actually has great power coupling efficiency, even at relatively high angles. This is Amanda moving the film around. Near field doesn't do quite as well. Ultrasound will also follow this similar trend because that's just the geometric properties of the angular misalignment. You don't get field focusing effects with ultrasound crystals. All right, so we took this idea, we worked with Kaiwan Yang at Rice, and we developed this combination of magnetoelectrics for wireless power and created our own ASIC as a neural stimulator. And the way this ASIC works is that we send power in with a magnetic field. We can also send data by modulating the field strength. So we can send a digital bit stream. We can tell the stimulator what stimulus amplitude and timing we want, and then we can put it into animals and see if it works. So here's an example of work that we did with Kava Kameri to show you that under realistic operating conditions, which I think is really important when we're baking these new technologies, that the device operates. So here's an example of a hemi-Parkinsonian rat model. And what happens is basically a phenotype for the disease is that the animal is discoordinated on one side of its body. It's basically, we give it a stimulant, it tries to run around, but due to the hemi-Parkinsonian lesion, one side is less coordinated, so it spins in a circle. But we know that deep brain stimulation works at about 130 hertz. And so when we turn our device on, and by turning on, I mean, we just tune the frequency of the magnetic field to the resonance. So if I'm off resonance, nothing really happens. If I'm on resonance, we see that we're really effective as a deep brain stimulator in this freely moving rat, right? I'm sending energy using this magnetic coil through air in a freely behaving animal. So that was actually, I think, a huge leap for Amanda's work, going all the way from concept to something that actually works in an animal. We've done some other studies to show place preference and things like that. We've now taken this a step further and thought about how we could actually develop a biomedical device. And that's work with Peter Kahn and Sunil Sheth. Peter is actually an endovascular neurosurgeon, so we're kind of taking steps toward making things that can be implanted through the vasculature on stents. But for the time being, this is placed on the nerve and powered remotely with a magnetic field. This is in a pig model. And so we're looking at this as kind of pain therapies. There's work that is just funded by NSF, and this was led by Jonathan Rivney at Northwestern, to take the same idea, create these miniaturized bioelectronic devices to help with nerve gap regeneration. So if we apply electrical stimulation, we can help guide nerve fibers to regenerate beyond a critical gap. And I think what's important here is that we're able to miniaturize the entire system, combine this with state-of-the-art hydrogels and conductive polymeric materials, flexible electronics, and kind of build this kind of bioelectronic integrated steps towards this distributed system of integrated bioelectronics. All right, let's see. So I have, I think, a little bit more time. So that's basically what I wanted to talk about in terms of magnetoelectrics. there's another direction that I want to go, which is kind of more towards this biohybrids that I had mentioned. So what we can do with the magnetoelectric materials is we can power an ASIC, and the ASIC can do basically CMOS-based bioelectronics. But sometimes I think it's important for us to think about more biochemical capabilities. And in that case, we've been thinking about how we can use magnetic fields to control cell activity directly, or to use engineered cells to create biomolecules. And so this is really inspired by work from Paulina Anakiva's lab and Arne Prowl's lab that showed that if you use magnetic nanoparticles, you can heat them up with alternating magnetic fields. And if those cells express thermoreceptors, you can activate those thermoreceptors and drive a biological process. This is either a change in the transmembrane potential or potentially activated gene circuit. So the idea is that nanoparticles next to genetically modified cells would activate those cells and only those cells. Now, an alternative is that we could create engineered cells and encapsulate them and protect them from the body's immune system and surround them with a nanoparticle shell. And so we could actually use these engineered cells to produce biomolecules under remote control for magnetic fields. And this is work with Omid Baisa and Gang Bao. Okay, great. So this seems like a solved problem, right? Magnetic fields heat nanoparticles, that's been shown. We can just kind of couple this with what's already existing. So let's see how this works in vivo. So this is work from Arne Prowl's lab. This is the state of the art in magnetogenetics. This is magnetogenetics because there's these cells are genetically modified so that the activity is targeted to select cells. So the state of the art in magnetogenetics is something like this. This animal has nanoparticles injected into half of the motor cortex. The field turns on. Those cells should be activated, and we're expecting to see the animal turn. But I'm still talking, and the animal hasn't turned yet. There it goes. Okay, so now it starts to spin in a circle, and note this video has been sped up 3 times. So the challenge here, I think, with magnetogenetics is that there's a long latency between when I turn my field on and the cells respond. We didn't see this with magnetoelectrics because I'm using an electronic stimulator. Basically, I turn on the electric field, the cells respond immediately. Using a thermal mechanism, we have these long latencies, which I think has been a major limitation in terms of how we can apply this technique. So we thought about this, and we thought that maybe the issue is that we need to think about this as a system where we can engineer both the nanoparticles and the applied magnetic field, as well as the thermoreceptors that transduce that heat. And so this realization that this is a system that needs to be designed, Charles Sebesta in my lab came up with the idea of using thermoreceptors from Drosophila, which have been shown to be very sensitive and sensitive to the rate of heating, which allows us to have much faster activation times. And here's an example. This fly has been genetically modified so that when we turn the magnetic field on, we are activating the neurons associated with this mating ritual where it like sticks one of its wings out. So you see when we turn the magnetic field on, flies do their mating dance. We turn the magnetic field off and the wings relax almost immediately. So this is about a 50x improvement in terms of the latency. We're getting sub-second response times compared to a 30-second response time that we had seen previously. And the insight here really from Charles is that by thinking about engineering the right thermoreceptors, we're able to actually expand the capabilities of this technique. So now we're working on these kind of hybrid systems that allow us to use these engineered cells with Omid Baisa and Phil Horner at HMRI to control the production of biomolecules like IL-10, for example, under remote control from this magnetic field. All right, so I think a hard time, not sure how much time I have here, but I have a few slides.
About 7 more minutes.
Seven more minutes, all right, plenty of time. Okay, so I have a few slides on actual optics, which I promised you guys I would talk about. Okay, so we've been thinking about magnetics, and I think where we found magnetics to be very powerful is on actuation. So we've been able to send power and data into the system, been able to activate nerves, control biomolecule production. Getting information back out with magnetic fields has been, turns out to be more challenging for us. And that's what, so where we see optics playing a role is getting this information back out. We know that we can genetically modify cells so that changes in calcium, which correspond to changes in voltage, can be converted into a fluorescent signal if they're genetically modified with this calcium-sensitive fluorescent proteins. We also know that with integrated photonic devices, we have the ability to perform a lot of processing to optical waves, similar to what we do with a microscope, but on a planar substrate. So it can control amplitude and phase of incident lights. We have great detectors and modulators, et cetera. And the thought is that if you can use this kind of platform technology combined with computational imaging, we would be able to build imaging systems or sensors that could be planar implanted, that have the same kind of form factor that we think of with implanted bioelectronic devices, and they don't have the traditional lens-based architecture that we're come to associate with microscopy. And so that was the idea that we started with. I began working with Ashok Viraghavan's group, who are really the experts in computational imaging. And in my lab, we had a background in integrative photonics and some biology, so we thought we could bring all these things together. Our first demonstration of this was the idea that we could build a microscope without any lenses that could be made completely flat. And the way that this works is that we use a mask, either a phase mask or an amplitude mask, to perform basically a computation on the incident wave front. So the incident wave front comes in, we have a known transfer function between this phase mask and our sensor. And that results in something that looks like this. It doesn't look much like an image, but there's information in this raw sensor capture. And that information is conditioned by our phase mask or amplitude mask. And if we do this conditioning right, we can invert this image or deconvolve this image in a way to create what looks like a microscope image. Here's an example of this. And one of the nice things about this is that because this is a completely computational process, this is one image capture that we can then computationally refocus until we recover an image of the surface of the brain that is close to what we would get with an epifluorescence microscope. It's not perfect and the resolution isn't quite as good as we get with fluorescence, but we have a big win here in terms of the form factor. We get some other wins too in terms of field of view, but that's a little bit niche. And the important thing here is that when we're thinking about these neural interfaces, we don't always want to form beautiful images, you just want to get the data out. And so what we've been able to do is we've been able to use this to measure calcium dynamics. This is a movie of the raw sensor capture, which doesn't look like much is really changing, but once we perform our reconstruction, you can see the flickering of the calcium dynamics inside the cortex, similar to what we see with epifluorescence microscopy. And again, in this kind of planar form factor. So we're excited about building this. This was actually funded by DARPA in their NESD program. It's kind of high bandwidth neural interface. And that's wrapping up, but we're hoping to be able to do move this into non-human primates by the end of the year. So we're excited about this as kind of an optical brain-computer interface. The other thing that we've been thinking about we can do with light, and Mason talked about this, is that maybe we can use light externally and get information from what's happening inside the brain through the skull, which is really hard for something like this. We would really not be able to do epifluorescence imaging through the skull because there's way too much scattering, but we might be able to extract information in other ways. And so the idea here is that if I try and send a photon through the skull, most of those photons would get scattered by the surface and basically bounce back. So if I send light in here and detect light over here, most of the photons are colored blue because they basically don't go into the brain. I'll color a few of these orange in this kind of region of interest. These are the photons that happen to bounce all the way through the skull, through the cerebral spinal fluid, into the brain, and bounce back out through my detector. So a very small fraction of those photons actually do that. But if I add temporal resolution to my detector, meaning that I send a pulse in and I look at the photons that took a long time to bounce back, and by long time, I mean like 200 picoseconds. So not a super long time, but a little bit longer than these early arriving photons. We see a lot of them actually do bounce through the surface of the brain. And so by basically using this time gating approach that we get with these single photon avalanche detectors, we can improve our signal to noise for this through skull imaging. And that's kind of the insight behind these kind of non-invasive neural interfaces that we're working on as part of DARPA's NCube program. And I'll show you some early results. This is a combination of measurement and simulation because we got shut down for COVID, but we're actually beginning to collect this data now. So what I'm showing you on the top is what we would get from an epifluorescence microscope. So this is the surface of the brain. These animals have this calcium sensitive indicator. And what I'm doing is I'm flashing one visual stimulus on the left side and the right side. And what we see is brain activation on the contralateral side, as you would expect. Here's a little movie. So this is what looks like with the epifluorescence microscope. It's really nice. You can see the vasculature and everything. Now, if I put a skull phantom or simulate the effect of a skull phantom, you'll see we lose that spatial resolution, as you would expect because of the scattering. Now what we can do is we can add the time of flight, and we can add some clever computational techniques from Ashok Viraghavan's group and Andreas Hilscher's group at Columbia. These are based on optical tomography, and maybe Jana's going to talk about these kinds of techniques later. We can do some source localization on the data combined with our time of flight measurements, and we get something with much better spatial resolution, and we can recover this kind of contralateral stimulation pattern. This distance is about 4 millimeters. So we're approaching this kind of 1 millimeter spatial resolution through the skull using light. And this is actually blue light that's being used in this experiment. And then we can do a lot better as we shift these kind of red-shifted indicators. So I think there's actually exciting opportunities to take some optical techniques and create non-invasive neural interfaces. This is, as I mentioned, this is part of DARPA's NCube program. So there's a ton of people who are doing awesome work on this project, but kind of the dream scenario is you have this magical helmet that you can wear to do stimulation of select neural circuits using the magnetic techniques that I had talked about, combined with optical techniques for the readout, so that we get this bidirectionality by using kind of a mixed mode approach. And so if I have maybe 2 minutes, I'll mention something that's slightly very unrelated, but I just like to talk about it. You know, a lot of our work is kind of focused toward human applications. So we're thinking about technologies where at most we're going to be able to sample from some tiny, tiny fraction of all the neurons in the human brain. But I think for neuroscience applications, you can also think about developing technologies at the other extreme, where we might be able to measure the activity of all of the neurons inside of an animal as it behaves. The advantage here is that I think we can develop a kind of rich understanding of brain behavior relationships because we have complete information. So as we've been looking at developing systems that allow us to do this in animals like worms and hydra. And we're going to borrow actually a lot of the same technologies, this microfabrication technologies, both for electronics and fluidics. And we're basically going to build neuroscience chambers on a chip. And so we've been doing that. I think it's a lot of fun. If you guys want to chat about it, I would be happy to tell you guys more. But we can basically put electrophysiology, imaging, as well as various types of stimuli, mechanical, chemical, and temperature stimuli, all on like a platform that's very scalable. So we can do massively parallel neuroscience experiments in these organisms. And here's a couple of examples of electrical recording and imaging, chemical stimulation. These are hydra. So you can see actually each one of these dots is a neuron. It's the whole animal as it moves around. You can basically, now there's a huge tracking problem, but if we saw that, we could actually watch all of the neurons in this animal during behaviors and chemical stimulation. We've developed ways to do thermal stimulation also. Again, if you want to follow up on any of this, there's a number of publications here over in the corner. And I always like to mention before I get a question from the audience about like why I'm creating brain interfaces for the government, that I think it's important for us to incorporate and kind of bake in ethical principles as we develop this technology. There's work with IEEE Brain. If you want to find out more, there's a website here. We're actually working with engineers and ethicists now to kind of fill out this kind of matrix of ethical considerations. And I think it's important that we, particularly for something that's as important as connecting to the human brain, that we develop a culture of including ethical principles as we're developing these technologies. And so I think we can all kind of come together on that. And if you're more interested, here's some stuff to follow up on. And that's basically it. Like I mentioned, there's a big, fantastic group of students who contributed to this. I'm also fortunate to be a part of the neuroengineering community at Rice. There's an initiative now that hired Chong Shi, Lan Luan, and Jerzy Zablowski last year. There's more faculty positions coming open, lots of student and postdoc positions, both in my lab and in a number of labs over here. We've just relocated to new lab space. So I think it's a really exciting time for us. And I think CMU also has a similar type of community. So I love coming and chatting with you guys because you bring a lot of similar ideas together. So it's been a pleasure to be a part of this. Thanks.
Thank you so much. Thank you so much, Jacob. There are 2 questions in Q&A and one from the panelists. I do want to take Pokit's question first because I had the same, which was for the 3D reconstruction with the time of flight DOT. Are you measuring the hemodynamics, I presume, or is it What signal are you looking at? Is it?
This is actually fluorescence.
Oh, okay. Okay.
So we're thinking about a similar approach should be able to do hemodynamics. For what we wanted to do with DARPA, we wanted really fast temporal resolution.
Right.
And we think that the hemodynamic response isn't going to approach sub-second temporal dynamics, but fluorescence will. And so we're looking at both voltage and calcium sensitive indicators.
Sorry, not
But
being an optics person, do you need to add something to the cell there?
Yeah, and so what we're working on there's a lot of stuff that I glossed over. So what we're working on is we're working with Elisa Conefogo's lab at Columbia to introduce viral vectors by injecting them into the bloodstream and then using ultrasound to disrupt the blood-brain barrier to deliver those vectors to the brain non-surgically. So you have to deliver a contrast, but we can do that with viral vectors.
That's awesome.
In mice, you can use transgenic animals or, yeah.
That is super awesome, yeah. I know Mason also has a question, but let me go to the one from the audience first. The first one from the audience was regarding the endovascular placement. What are the precautions that will be taken to prevent thrombosis and or stenosis? What will be the potential applications of endovascular devices?
Yeah, great question. So based on the conversation I've had with surgeons, there's kind of two approaches. One is to target large vessels and ensure that our device is very thin. In the case of stents that are thin in these large vessels, they tend to be endothelialized. So the vessel will kind of grow over your device and it'll become a part of the vessel wall, in which case the risk of thrombosis is low. It's possible you might have to be on antiplatelet therapy or something like that, but we think for the large vessels that won't be the case. For small vessels and other targets, what we're thinking about is there are places in the body where vessels are often cauterized during surgery to prevent bleeding. And so we're thinking it might be possible to deliver our device to one of the vessels that are going to be cauterized, dropping it off, cauterizing the vessel so it's no longer part of the vasculature. We're just using the vasculature as an entry point to deliver those devices.
Okay. One more question from the audience before Mason, sorry. There was a question about for the magnetoelectric devices, are you considering going towards higher frequencies in order to reduce the size of the implant further, such as microfabricated magnetoelectric devices?
Yeah, we've looked at some of the microfabricated magnetoelectric work. Nyansun's lab at Northeastern has done some really good work in that area. We think that the right size scale for us is going to be on the order of millimeters. And the reason why is that for these very small devices, you don't get great, you don't generate a lot of power. We don't think we're going to generate enough power to do the kinds of things that we want to do. At the millimeter size, we're generating milliwatt levels and maybe several hundred microwatts of usable power. That seems to be good for a lot of the stuff that we do. So I think that, and it's also in that size range where we could inject it or implant it endovascularly. So I think the sacrifice that you get in terms of total power generation really is a setback for the microfabricated stuff. Also the high frequencies above 500 kilohertz, that's kind of our target range. Once you get above 500 kilohertz, you actually start to have some heating of the tissue due to the eddy currents. And so I think that there's a challenge there at the gigahertz range for magnetic fields. So those are the two reasons why I've stayed away from the micro.
I see. Mason?
Thank you very much, Jacob, for the really exciting talk. So I have a general question. I mean, for example, for your magnetoelectric technique, I assume there will be some energy loss when you transform energy from one domain to the other and then to the other domain, right?
Yeah.
But then on the other hand, you are gaining in terms of propagation loss and maybe insertion loss to couple energy to your device. So I was wondering, how do these confounding effects play when we think about energy conversion in your devices?
Yeah, this is why I don't love this way of drawing it, but it's the best I could, because when you bring up that there's actually many factors. I think one of the factors that you're alluding to is that, you know, there's an efficiency factor. And we actually have poor efficiency compared to ultrasound for two reasons. One is that we're not focusing energy because we don't have a propagating wave. So we're basically we're blasting magnetic field everywhere, and we're only picking it up in a small area. But, you know, and you also mentioned that we have some loss here. So there's kind of two efficiency terms that we're hurt by. We think that is okay for many applications because on the transmitter side, you actually have a pretty high power budget. I can turn up the power on my transmitter up to the safety limit. And so then the real question is, how much power can I safely transmit? And then what is my efficiency? And then how does that lead to my total power of my device? And it turns out at the frequencies that we're working with magnetic fields, the safety limit is quite high. We're very far from the safety limits. So we can crank that field up by about an order of magnitude before we start to reach safety limits. And this is a challenge with ultrasound, because ultrasound is actually, for some applications, gets close to that safety limit. So you really can't bump the transmitter power up to compensate for low efficiency. And so I think what we benefit from is the idea that we can bump up our transmitter power and to get total delivered power up high, even if we suffer from low efficiency. Now, this isn't a completely, this trade-off hurts us in a sense that if you want like a battery powered device, our battery lifetime suffers as we crank the power life up. So I mean, there's lots of, basically you have to do a system design in order to approach your solution, but those are the trade-offs that I think are important.
I see. And is there any possibility to focus the magnetic field, or?
There, you know, I hesitate to say no, but I think that that's very, very challenging to do. You don't get the interference effect that you get with the propagating wave. You know. Maybe you can come up with something clever with ultrasound or something.
Thanks, Jacob. I'm sure Mason, you can ask more questions in the chat. If anyone else has questions for Jacob, please ask them in the Q&A. We do need to move on to stay on schedule. So I feel kind of bad cutting you off since I'm the next speaker. But yeah.
I'm introducing you. So yeah, sorry, my question took long. So it's a great pleasure for me to introduce the next speaker of the session, Professor Jana Keinersdorfer. Jana is an associate professor of biomedical engineering at Carnegie Mellon. She holds courtesy appointments in Neuroscience Institute as well as the ECE department. Her lab is focused on developing non-invasive optical imaging methods for disease detection as well as treatment monitoring, with an emphasis on diffuse optical imaging.